Hello everyone, this is Dr.M.A.Bara from Department of Botany, Bharati Vishwavidyalaya. As we last see series on data analysis, right now today we will see what is biostatistic, and this is an important tool for data analysis. It is the next step on data analysis. Though this is whole different topic, biostatistic or bioinformatics is little bit different, though they are the part of maths. Mathematical tool or software or bioinformatics used to detail or develop program to analyze the functions of biology, any field of biology or any medical, biological or any kind of biological methodology to detail it to develop the software. All this included in biosystematic. So what is biosystematic? As we see, uh, verbal definition, as we see the verbal definition of biosystematic is the application of statistical technology to scientific research in health and relation related to scientific research or research in medical field or any kind of analysis, any to analyze any kind of methodology or experimental data you did in your science work or experiment data. Okay, it could be from agriculture, medical or any type of field. The most important to develop any tools in this area. Suppose we analyze the variables in the study, or these variables could be anything, that could be altitude, geospatial work, or population, community dynamics in plants. In plant science, mostly we deal with all the three areas. If we are doing about the geographical ecological work, we will study about the community dynamics, community. And if we see the work in medical field or pharma field, so it will deal with your computational work in bioinformatics, medical tool we need, or it could be biogeographical, that is altitudinal work or any type of work. These work are done on the basis of the bioinformatics tool developed in software program. We can use the platform as SPSS or any kind of analog or analog platform or any type of software that use to fragment out genomics, proteomics. There are many software we use to analyze the protein sequence, or it could be the different type of protein or other type of bio biomolecules, other type of biomolecules, or any type of compound related to blood or any type of sample. Okay, so basically the bioinformatics has two portion. First phase, to develop and data analysis with the help of statistics, and the second phase to use of methodology of biology or any type of field, any interdisciplinary field from where you collected the data. So these are the two different ways in which biosystematics works. So it will use any type of software. For example, if you want to do genetic analysis, you will approach a tree, or you can analyze it for RT-PCR, as RT-PCR is so famous right now. Everyone knows about RT-PCR test. It is used earlier. It is used to detect the family or genetic material, that is nucleotide. So in RT-PCR we double the genetic nucleotide, and it could be analyzed through analyzer which type of nucleotide is present in particular sample. So RT-PCR is one of the tool which is used, and which is used in bioinformatics. So you have a common example during this SARS outbreak, SARS-19, that is COVID-19. You people all know that RT-PCR, how it is sample is collected in biological terms or in medical terms. In medical discipline, you will see the samples were collected through nose, mouth, or sputum, or any kind of sample, and that it is used in within a 24 hours. The database is connected to your with your Aadhaar number and various things. These tools are one of the most strongest tool. Right now, it is proven during the pandemic what is biostatistic can do, the formulation or making vaccination, and the thing, and the sample were collected widely, vastly throughout the country, different region of the country, and all these with the help of the stata, we came to know where the pandemic is more, where the corona outbreak break or pandemic outbreak is more, where the sample is more collected, and where the disease are in endemic or pandemic stage. So this is a one of the example. We I'm going to see going to illustrate how this biostatistic tool, especially BLAST and different type of software developed by NCBI, how it is going to work. So the what the software they develop, what they do, they will collect the information with your bioassay. Bioassay, anything that is collected as your sample, it is analyzed with the methodology. फॉर एग्जाम्पल के हम देखेंगे बायोस से किसी भी सैंपल को कलेक्ट करना, जैसे पहले आप ब्लड सैंपल कलेक्ट करते थे, उसे स्लाइड में काउंट किया जाता था। आजकल क्या हो गया है, मशीन हो गया, यू कैन यूज रेडिली ईच एंड एवरीथिंग। सपोज इफ यू वांट टू डू शुगर टेस्ट, you will put a drop of blood, and that instrument, the blood sampler or the sugar test analyzer, will instant give you the reading of sugar. That is the one of the most handy use of biotechnology or biostatistic. Okay, so how that will produce the result, that strip has diode, and they count the sugar value from your blood, and they can give the interpretation of inside that chip. It will process and that produce some data, some out, they give some output in the numeric form, and that is the one of the easiest method of biostatistic. So you can while writing your answers or while interpretation what is biostatistic, you can give this example of both RT-PCR and sugar test of using sugar portable test tool. So how these efficient biostatistic tool are nowadays working. So first thing, most necessary is bioassay. Bioassay to analyze and what is ये जो bioassay का वर्क है, एरे जो आप साइंटिफिक डेटा एनालिस करते वक्त या स्टैटिस्टिकल या मैथ्स में आपने एक वर्ड यूज किया एरे, एंड द सिमिलर टाइप ऑफ वर्ड इज एसे. ओके, एलाइजा, एलाइजा इज ELISA, ELISA is enzyme linked amino assay. This is a word given to in which you can assay or you can check with which type of antibiotic is given in the sample. Okay, so this is another tool you can see on the screen that is preferable on RT-PCR. So, but this is a little costlier. That is why ELISA is not used worldwide for detection of COVID-19 samples. So here I want to say that different type of machine or tools. Okay, there are instrument in which you can put your sample that will give a range of your data. Okay, आप मशीन में अपना ब्लड सैंपल डाल सकते हैं और उसमें आपको ये पता चल जाएगी कि एंटीबॉडीज, एंटी सीरम आपके कितने ब्लड लेवल में क्या क्या डिफिशियंसी है. जैसे आपने आजकल बहुत सारे न्यूज़पेपर एडवर्टाइजमेंट में देखा है कि आप 600 रुपये में इंस्ट्रूमेंटेशन करा लीजिए, सीरोलॉजिकल डिटेक्शन करा लीजिए, यानी ब्लड कंपोनेंट के बहुत सारे टेस्ट करा लीजिए. सो हाउ दिस वर्क, दिस इज वन ऑफ द पार्ट ऑफ बायोस्टैटिस्टिक. Assaying any type of detection of any sample is your data processing or data collection. So there is three part: development, data analysis, and मेथोडोलॉजी ऑफ बायोलॉजिकल टूल. तो अगर आप साइंस बैकग्राउंड से हैं, बायोलॉजिकल और लाइफ साइंस और बॉटनिकल साइंस से हैं, तो आप सैंपल कलेक्ट करेंगे, आप लीफ के पिगमेंट कलेक्ट करेंगे या ब्लड सैंपल कलेक्ट करेंगे और उसे आप RT-PCR में डाल देंगे. इफ इट इज़ अ DNA और द सैंपल कुड बी एनीथिंग, इट कुड बी DNA, it could be protein, it could be any type of serological piece of your work. ठीक है, आपके वर्क के अकॉर्डिंग सैंपल चेंज होते रहेंगे. So what you will do, you will collect and you will put in analyzer. जैसे फर्स्ट RT-PCR में उसे डाला गया, वो रिपीट सीक्वेंस बनाएगा और उस रिपीट सीक्वेंस से फिर वो एनालाइजर में, मैं स्क्रीन पे हाउ न्यूक्लियोटाइड सीक्वेंस विल अपीयर एंड दीज न्यूक्लियोटाइड सीक्वेंस A to G and G to C on the basis of G to C ratio, it will collect the data and it will give the interface कि ये इस चीज को belong करता है. Same thing happen in criminology or forensic, how a small amount of hair follicle will give the test of which person, which parents this person was belong to. In forensic, we so if we solve murder mysteries or any homicides, so we can give these type of technology will give answer to them. So why in research methodology, why it is necessary, we will develop any kind of work or any kind of result. First foremost thing, this is a part of data analysis. Second thing, if you have been being a researcher or research scholar, you have to you should have knowledge about which type of tools and techniques are currently used or currently what are the most advanced technologies used by researchers or in science world. And third, if you want to develop or you want to work proceed in the research field, you must know which type of tools and techniques are used. So bioinformatics has the outcome of on three phases. So what is the necessary step to design any software or data system, and collecting data analysis, the second one, and the third one interpreting of these samples. Okay, so bioinformatics will develop a database, and how this database work we will see on this screen. Here the sequence number, the protein design. First on this figure you will easily see there is a common tree. It is developed with tree. Tree is a software which is designed by NCBI, and on the nucleotide sequence it will give the analyze give which this species is belong to this or that family. You can see here common tree, taxonomical report, and 3D structure COD on the protein. Accession number is given in the right hand side. आप किसी भी तरह का सैंपल जब आप जमा करते हैं, आप न्यूक्लियोटाइड सीक्वेंस, these portals are work on online. Okay, these portal you have to submit your nucleotide sequence in this website, and they will analyze it readily, and they will analyze and they will give the accession number, and then there is a G1 ID score card. There is a score. मैंने आपको स्कोर को बताया था. कभी भी क्या होता है, जो हमारे पास प्लांट है, सपोज हमारे पास कोई प्लांट सैंपल है, हमने उसका DNA आइसोलेशन किया, RT-PCR किया, प्रोटीन एनालाइज किया, और उसके बाद हमें क्या मिलता है, न्यूक्लियोटाइड सीक्वेंस मिलता है. आप अगर ब्लड सैंपल से भी कुछ चीज कर रहे हैं, किसी डेड बॉडी, suppose in forensic science if you found some dead body, you have such you cannot recognize that corpse. So what you will do, आप उस डेड बॉडी से थोड़ा सा कुछ सैंपल उठाएंगे और उसको आप एनालाइज करेंगे. कैसे एनालाइज करेंगे, उसकी जेनेटिक कोड निकालेंगे, और जेनेटिक कोड के लिए बायोलॉजिकल मेथोडोलॉजी यूज करेंगे. डीएनए आइसोलेशन के लिए बायोलॉजिकल मेथोडोलॉजी यूज करेंगे. मोस्ट ऑफ द टाइम CTAB मेथड, and there is an OH method. There are so much methodology are there. So you will use any of one of that methodology, and you will develop you will bring out the genetic code, and you will put in this online software, and this will analyze. This is a data this is a database where any record or any record is found to match the plant. तो हम plant के cases में या zoological, क्योंकि ये एक IUCN NCBI से regulated है, तो जो भी हमारे plant के data है, हम paper publish करते हैं या किसी भी तरह का analyze करते हैं, या कोई संस्था बहुत सारे plants को recognize कराती है, तो उसका database यहाँ पे किसी भी NCBI जैसी संस्थाओं में attach करती रहती है. और उसके बाद IUCN सब जानते हैं, बायोडाइवर्सिटी की जो रेड डेटा बुक लिस्ट है, उसकी जो हेड है IUCN, वो इन सभी टाइप के जेनेटिक इंफॉर्मेशन का डेटाबेस कलेक्ट करती है, और वो इस तरह के पोर्टल्स में इसको डालती है. तो वो क्या होता है, जब भी कोई नया प्लांट आपको मिलता है, या यू विल फाइंड एनी न्यू स्पीशीज, दे विल गिव अ स्कोर कार्ड. दे हैव some software online portal in which you will put your nucleotide sequence, and the outcome will comes under the score. वो कितना जो पहले प्लांट सैंपल है उससे कितना मैच करती है. If there is no match, or if there is 95 less than match of 95%, जैसे आपके पास अमलताश है और आपके पास अमलताश जैसे दिखने वाला एक और पौधा है जिसकी पत्तियां थोड़ी छोटी है, आप उसका सीक्वेंस अगर आपको डाउट है, हर तरह से आपने उसको मैच करके देख लिया, दोनों एक ही पौधे लगे, फिर भी आपको डाउट है कि ये दूसरी वेराइटी है, ये दूसरी स्पीशीज है, you can find out its genetic code, and you will submit this into your you will do BLAST, and then this will give out a sequence on the basis of protein. As you know, nucleotide is nothing but protein. So this is how the any type of protein or any type of sequencing is recognized on the website or NCBI link. Okay, and this will give our complete data. You can download that and you can publish, use this data on your use this data and you can publish your paper. It is fully recognizable, and it is total authenticated. Okay, so NCBI Blast, these platform are developed on the basis of language, that is Python, BioPython 71. What they do, they have environmental data, literature and NCDI. As आपको एक और वेबसाइट के बारे में पता है, पबमेड, उनके पास तीनों चीज़ों के डेटा कंपाइल्ड है, डेटाबेस भी है, जो लिटरेचर पब्लिश्ड है वो भी है, और प्लस जो भी नया संस्थाओं से जो कुछ भी डेटा है, जैसे इंडिया में एनसीबीआई है, ब्रिटेन में क्यू बॉटनिकल गार्डन है, तो उनके पास जो भी फिजिकल डेटा भी है. ओके, पबमेड है, इफ दे आर पब्लिशिंग एनीथिंग और दे आर अंडरटेकिंग द पब्लिशिंग हाउस. ओके, किसी एक जैसे साइंस डायरेक्ट के अंदर बहुत, एल्सवेयर के अंदर अदर बहुत बहुत डिफरेंट टाइप ऑफ पब्लिकेशन है, तो दैट इज अ पब्लिशिंग हाउस. दे हैव many journals, they have their own data, so they will collect, that is MBGBDB, as it is shown in diagram. It has meta storage and Oceanica database. Okay, they have two different type, the data themselves and the data they are generated by the cross linkage, mm, cross collection of other literature, or they will see all these and they match their genetic portion, and then they will generate a result. So how this is biostatistical things work, these are very much clear with this figure, how biostatistic work and what is the platform. And this is the NCBI website, web page, where you can put all your resources. Here is a screenshot of different type of tab. If you will land there NCBI website, you will find all these different tab, blast sequence for different for each and every type of. If you want a literature, there is a different tab, there is different button, and homology, proteins, if you want to identify any plant with the help of protein, if you want to sequence, you want to analyze any plant material with sequence, or you have taxonomy or training tutorials or anything, with NCBI has its with its database, you can access through. So with the same thing, if you blast it, okay, NCBI provide a search engine through which you can match the plant sequence, but it will also access your data also. If you had something new, you have to submit it and it will give your accession number. That is how you will authenticate it, and this work is vice versa. You have to submit your data and they will provide your accession number, and this is how your work will be authenticated it, and how database is developed and designed. So interpreting your data, that is nucleotide sequence, it will give outcome in such way. Here you can see the sequencing and how scorecard are, uh, the red lines are the similarity between your data and the already data they had. So uh, on these platform, the BioPython platform, NCBI will produce all the tools, all your the technique you want to access. So this is for your biostatistic. Biostatistic, in simple ways we can say, different type of tools is developed to resolve your biological methodology. Okay, so uh, be done with this biostatistic class. We will meet in next lecture. Thank you.